Very good morning, class. So today we'll be continuing about dental plaque as a biofilm. So today we'll be discussing about the various theories of dental plaque formation. So before we discuss about theories, we'll just briefly discuss about what happened in the previous class. So how this plaque gets attached to the tooth surface is by transport to the surface. The transport of the bacterium to the tooth surface happens through three motions. The first one is Brownian motion, and the second one is sedimentation, and the third one is chemotactic activity. The second phase is initial adhesion. The second stage results in initial reversible adhesion of the bacterium. This is initiated when the bacterial cells comes into close proximity with the surface and through three forces. The first one is long force. Second one is short force, which includes van der Waals forces and electrostatic repulsive forces. The electrostatic repulsive forces prevents bacterial cells from being even closer to the surface. So it is thought that a stronger binding at this point is the consequence of interactions between the bacterial adhesins and receptors in the salivary pellicle. The third phase is strong attachment. So after initial adhesion, a firm anchorage between the bacterium and the surface is established. So on the rough surface, the bacteria are more protected against shear forces, which changes from reversible to irreversible binding, which occur more easily and more frequently. This binding between the bacteria and the pellicle is mediated by specific adhesins on the bacterial cell surface and the complementary receptors like proteins, glycoproteins, or polysaccharides in the acquired pellicle. These proteins in the acquired pellicle act as receptors for streptococci, including alpha amylase, acid proline-rich proteins, stadherin, and salivary agglutinin glycoproteins. The specific adhesins of primary colonizing bacteria have been the subject of many investigations because these represent potential targets for interference with the formation of dental biofilm. So how this colonization and biofilm maturation happens? The primary colonizing bacteria adheres to the tooth surface and provides new receptors for attachment by other bacteria as a part of process known as co-adhesion. This co-adhesion leads to development of microcolonies. There is a cell-to-cell adhesion taking place genetically between distinct oral bacteria, which occurs in fluid phase. So there are three theories in plaque formation. The first one is non-specific plaque hypothesis. In this hypothesis, it maintains the periodontal disease resulting from the elaboration of noxious products by the entire plaque flora. According to this theory, when only small amount of plaque are present, noxious products are neutralized by the host, and the large amount of plaque would produce large amount of noxious products, which would essentially overwhelm the host defenses. So in this, the concept is that the control of periodontal disease depends on the control of the amount of plaque accumulation. Treatment of periodontitis by debridement by surgical or non-surgical and oral hygiene measures focuses on the removal of plaque and its products and is founded in the non-specific plaque hypothesis. The observations of this hypothesis were some individuals with considerable amount of plaque and calculus and gingivitis with never developed destructive periodontitis. Individuals with periodontitis showed site specificity in disease pattern. That is, some sites remain unaffected, whereas some sites showed advanced disease. So with the concept that all plaque was equally pathogenic and the presence of uniform host defenses, these findings were inconsistent. The specific plaque hypothesis. This hypothesis explains the pathogenicity of dental biofilm depends on the presence of or an increase in specific microorganisms. This concept encapsulates that biofilms harboring specific pathogens provoke periodontal disease because key organisms produce substances which mediates the destruction of host diseases. Ecologic plague hypothesis. During 1990s, Marsh and co-workers developed the ecologic plague hypothesis to unify the existing theories regarding the role of dental biofilms in oral disease. The principle of this hypothesis are that both the total amount of dental biofilm and the specific microbial composition of biofilms may contribute to the transition from health to disease. So the health associated dental microbiota is considered to be relatively stable over time and in a state of dynamic equilibrium or microbial homeostasis. So the host controls subgingival biofilms to some extent by a tempered immune response and low level of GCF flow. So a change in the nutrient status of a pocket or chemical and physical changes to the habitat are considered the primary cause for the overgrowth by the pathogens. Disease could be controlled not only by targeting putative pathogens, but also by interfering with factors responsible for driving the deleterious shifts in the microflora by reducing GCF rate or site could be made less anaerobic by use of redox agents. The key elements of both specific and non-specific plague hypothesis are the selection of pathogenic bacteria is directly coupled to changes in the environment. Diseases need not to have a specific etiology. Any species with relevant trait can contribute to the disease process. So what is a biofilm? Biofilm refers to the relatively undefinable microbial community associated with the tooth surface or any other hard, non-shedding material randomly distributed in a shaped matrix or glycocalyx. There are two types of layers, lower layer and loose layer. Lower layer, these are dense microbes which are bound together in a polysaccharide matrix with other organic and inorganic material. And the loose layer is a loose, irregular layer extending into the surrounding medium. This is suspended in a fluid medium bordering the biofilm and act as a stationary sublayer and a fluid layer in motion. Properties of biofilm: cooperating community of various types of microorganisms. Microorganisms are arranged in microcolonies with channels between the microcolonies, and microcolonies are surrounded by protective matrix. These are differing environment within the microcolonies in the biofilm. Microbial gene expression differs when microorganisms are in a biofilm. Microorganisms have primitive communication system, and the microorganisms in biofilm are resistant to antibiotics, antimicrobials, and host response. Dental plaque represents a true biofilm which consists of bacteria in extracellular matrix composed of bacterial polymers and saliva or gingival exudate products. What is meant by quorum sensing? Quorum sensing in bacteria involves the regulation of expression of specific genes through the accumulation of signaling compounds that mediate intercellular communication. This totally depends on cell density. With few cells, signaling compounds may be produced at low levels. However, in autoinduction, leading to increased concentration as cell density increases. So once the signaling compounds reach a threshold level, gene expression is activated. So when this gene expression is activated, there is expression of genes for antibiotic resistance at high cell densities, which provide protection to the bacteria. And this also has the potential to influence community structure by encouraging the growth of beneficial species and discouraging the growth of competitors. It also plays an important role as physiological properties of bacteria and community is altered through quorum sensing. And the possible role of quorum sensing is influencing the properties of biofilm was first explained by Cooper et al. In 1995. The characteristics of supra and subgingival biofilms. The location of supragingival biofilm, as the name indicates, it is coronal to the margin of free gingiva. The origin of supragingival biofilm is salivary glycoprotein forming pellicle. Microorganisms from the saliva are selectively attracted to the pellicle. It is distributed on the proximal surfaces and the areas where the patient neglects, and cervical third and lingual mandibular molars, proximal surfaces, pit and fissure biofilms. It is firmly attached to the acquired pellicle and tooth surface. It is retention of supragingival biofilm is on the rough surfaces of teeth or restorations, malpositioned teeth, and carious lesion. The structure of supragingival biofilm is densely packed microbial layer over pellicle on tooth surface. And these are small isolated colonies forming between 2 to 5 days. And the colonies merge to form a covering of biofilm. The microorganisms are gram-positive cocci And in older biofilm, these microorganisms are filaments and fusiforms. The subgingival biofilm, the origin is apical to the margin of free gingiva. The location is apical to the margin of free gingiva, and the origin is downgrowth of bacteria from supragingival biofilm. It is distributed in shallow pockets, and and the biofilm is attached over the calculus. It is directly attached to the tooth surface, and the subgingival biofilm and the calculus. These are loose floating motile organisms in deep pockets, which do not directly adhere to the biofilm and to the tooth surface and pocket epithelium. The retention of subgingival biofilm is pocket holding biofilm against the tooth, and the overhanging restorations extending into the pockets. There are three layers: tooth surface attached biofilm, gram-positive rods and cocci, unattached biofilm in the middle, which consists of gram-negative motile forms and spirochetes. And the third one is epithelial attached biofilm. These are gram-negative motile forms predominating many leukocytes migrating through epithelium. And the environment conducive to the growth of anaerobic population. The diseased pocket is mainly consist of primarily gram-negative motile spirochetes and rods. The sources of nutrition of supragingival is from saliva and ingested food, and the subgingival plaque is from tissue fluid, exudate, and leukocytes. Supragingival biofilm is responsible for development of gingivitis, supragingival calculus, and dental caries, and subgingival biofilm is responsible for development of gingivitis, periodontal infections, and subgingival calculus. The benefits of dental plaque is a part of the natural resident microflora of the human body, and it plays a critical role in normal development of physiology of the host. The germ-free animals have altered mucosal surfaces, poor nutrient absorption, and suffering from nutrient deficiencies, and have impaired host defenses. So these resident microflora also reduces the risk of infection by acting as a barrier to colonization by exogenous and often pathogenic species termed as colonization resistance. Association of plaque microorganisms with periodontal diseases. The factors responsible are the susceptible host, the presence of pathogenic species, and the absence or small proportion of beneficial bacteria. The small amount of plaque of bacterial plaque can be controlled by body's defense mechanisms without destruction, but the balance between the bacterial load and host response is disturbed, leading to periodontal destruction. And the susceptibility of the host is partially hereditary, which can be influenced by environmental and behavioral factors such as smoking, stress, and diabetes. The second essential factor for disease initiation and progression is the presence of one or more pathogens of susceptible clonal type and in insufficient numbers. A. comitans T for Zythia P. gingivalis are key pathogens because they are strongly associated with periodontal disease and progression. Cox postulates, according to Robert Koch in 1870, he developed a basic criteria of microorganisms which are causative agents in human infections. These are: must be routinely isolated from diseased individuals, must be grown in pure culture in the laboratory, must produce a similar disease when inoculated into susceptible laboratory animals and must be recovered from lesions in a diseased laboratory animal. In the case of periodontitis, three primary problems were arising. The first one is the inability of the culture when the organisms that have been associated with the disease. And the second problem was difficulty in defining and culture sites of the active diseases. And the third one is lack of animal model system for the study of periodontitis. So Sokransky modified the factors for identifying the periodontal pathogens. So he described the criteria. The first one is must be associated with the disease as evident by increasing in the number of organisms at disease sites. Must be eliminated or decreased in site that demonstrate clinical resolution of disease with treatment. Must demonstrate a host response in the form of an alteration in the host cellular or humoral immune response. Must be capable of causing disease in experimental animal models. Must demonstrate virulence factors responsible for enabling the microorganism to cause destruction of the periodontal tissue. The two periodontal pathogens that have most thoroughly fulfilled Sokransky criteria are Actinobacillus actinomycetum comitans in the form of periodontal disease known as localized juvenile periodontitis and Porphyromonas gingivalis in the form of periodontal disease known as adult periodontitis. The treatment of periodontal biofilm can be categorized into three groups. The first one is physically remove microorganisms through mechanical debridement. The second one is to attempt to kill or affect the metabolism of the organism, which is antiseptics and antibiotics. And the third one is affecting the environment of the microorganisms. The newer strategies of controlling this plaque as a biofilm are the first one is control of nutrients, which is addition of base generating nutrients like arginine, reduction of GCF flow through anti-inflammatory agents, and inhibition of key microbial enzymes. The control of biofilm pH, which can be done by sugar substitutes, antimicrobial agents, fluoride, and stimulate base production. Control of redox potential by redox agents and oxygenating agents. And the other strategies are interfering with communication networks, prevent colonization of selected organisms, enzymes to dissolve matrix of biofilm, replace pathogens with a less virulent strain, and photoactivation of the microorganisms. We'll briefly talk about the microbiota of other periodontal conditions. In necrotizing periodontal diseases, what are the various bacteria we will be able to see? That one is fusiform bacteria, P. intermedia, Treponema selenomonas, and Fusobacterium species. In periodontal abscess, the mainly found classic periodontal pathogens commonly found are P. gingivalis, P. intermedia, P. melanogenica, F. nucleatum, T. forsythia, and Treponema species. In endoperiolesions, lesions, there are acute or chronic clinical conditions involving both the pulp and periodontal tissues. The two main signs and symptoms associated with the tooth affected by endoperio are deep periodontal pockets reaching or close to the apex and negative or altered response to the pulp. So the organisms involved are P. gingivalis, T. forsythia, P. micra, and species from genera Fusobacterium, Prevotella, and Treponema. In periimplantitis is a pathological condition in tissues around implants characterized by inflammation in the periimplant mucosa, and there is a progressive loss of supporting bone. So the organisms involved are P. gingivalis, T. denticola, and T. forsythia. And additionally, P. intermedia and C. rectus are also involved. So overall, we can conclude that the plaque as a biofilm, the treatments involved are oral hygiene measures, debridement by scaling and root planing, which is oriented towards the reducing the accumulation of plaque on the teeth. And the future development will be involving the development of therapies which prevent the colonization or growth of specific microorganisms that are known to function as pathogens in this environment. So with this, we'll conclude plaque as a biofilm. Thank you, everyone.